Okay. So good afternoon, everyone. Thank you for this opportunity to talk with you today. So my name is Etianne Martini Sasso. I'm also a PhD candidate from NCNED, and today I will speak with you about the TRPM3 IO channel, and as it is a potential pharmacotherapeutic target also in AMI and long COVID.
Sorry, Etienne, you just got on mute.
Yeah, can you hear me now?
Yep, that's all good.
Okay. Can you see my mouse moving or not?
Yes, yep.
Okay, great. Yeah, because I want to point some things. So first, I would like to talk about what is IO channel, okay? Because I will talk all the time about IO channel, so it's essential everyone understand what it is. So here I have a model cell. So this is the plasma membrane, and we have some IO channels here in different colors. So I would like to explain this like a door or a window in a house. So for example, in this other image, I have doors, windows. So think the IO channels are like doors or windows that I open and close. So for example, in our homes, when we want a fresh air, we open and it's allowed the air refresh in the house. Or when I buy some food, something for my family, I have to use the door to enter in the home and provide this for my family. So think the same way. When we have elements, we need these doors in the plasma membrane to open, and so I can use the calcium inside the cells for many functions, for example. So as Stanley mentioned before his presentation about some IO channels NCNED is looking, one example is the TRPM3 that they work, but it's from a big family of the Transient Receptor Potential IO channels, the TRPM channels. This superfamily have 28 members, and these members they are related to threats, stress, or all the other kind of stimulation that they responded to. For example, it could be infection, as happened with long COVID patient and many of the AM patients, and also could be some chemicals exposure, also can be differences in temperature and other examples. So most of these TRP channels are selective for calcium, what have this essential function for all different kind of cells. And important here also is the distribution. TRP channel have a distribution that is aligned with the AMI and long COVID symptoms. Sorry, I don't know. Yeah, it's gone. So specific about the TRPM3, NCNED is looking other TRP channels, but as my PhD is focused on TRPM3, I will focus in this presentation just in this IO channel. It's a non-selective high permeable calcium IO channel, and I'd like to explain about two drugs I use in the lab. So I use an agonist that is Progress. Progress is the one that will stimulate the IO channel, what make the IO channel open and permit calcium entry into the cell. Okay, and the second drug I apply in my experiment is ononetine, that is an antagonist. It's work closing the doors, close the IO channels, and reduce the current in these cells. So I don't know why it's not going for the next slide. Yeah. So here, TRP channels and natural killer cells. Natural killer cells as a model, NCNED have been used. So just explain basically, this blue one and purple here is the natural killer cells. These cells have this name because have the natural ability to kill target cells. Okay, so here you can see an example of target cells, and these target cells are like cells that are not good for us. So for example, we have infected cells, we have cancer cells, and other examples. I would say bad cells for us, that natural killer cells will naturally identify these bad cells and kill the cells, eliminate this from our organism. And these natural killer cells have the TRPM3 channels, not just one as represented here, but many different ones, TRPM3 ion channels in the plasma membrane. So what NCNED looked before was that there are important reduction in natural killer cell cytotoxicity in patients with MCFS. It was important because this is the function of the cells. So these patients not eliminate the target cells as expected, and also constantly reporting a significant reduction in the calcium mobilization. So there is less calcium inside the natural killer cells from M patients, and also reduction in the TRPM3 surface expression in MCFS patients. So the technique I use in the lab is the whole cell patch clamp. There are different techniques to study ion channel, but this is the gold standard. And basically, we have a microscope, and in this microscope, after isolate natural killer cells, it's important to highlight that natural killer cells are cells in our blood, so it's easy to isolate, it's easy to collect. It's not an invasive technique like a biopsy. And so after isolation of the cells, we look under microscope, and we use a kind of needle that is a glass pipette, and we select a good cell, and when you approach the cells, we apply a suction that will break the membrane. So if you look here, we have again the membrane, and we have the doors and windows, the ion channels in red or pink here. And this glass, when open the plasma membrane, I can record all the current that happen in the cell. So I apply the drugs I mentioned before, and I see if there are calcium entering or leaving the cells. So here's some results. First, with health controls and MCFS patients. So in health controls, what is expected is that we have a baseline, and when I start applying the drug that open the door, the progress, there are an increase in the current. It's the expected. And if you look in black here is my baseline, and after application of progress in red, you can see an important increase in current, what means that more calcium is entering the cells. However, when I look at M patients, there are no difference after application of progress. There are no response. The ion channel are not open properly. So when look statistically and compare all cells from all M and health control analyzed in this project, there are significant reduction in the amplitude of progress in compare health controls with M patients. What mean the ion channel is impaired or dysfunctional. We use the second drug to close, to block the ion channel, just to confirm. And in health controls, we can clearly see a decrease in current. Here, my baseline that now is progress, and there are reduction this time because the door is closing, so not more calcium entering to the cell. And again, when I look at ME patients, I have no difference. There are no decrease on anything not to closing the doors. It's also because progress didn't open these doors. Okay, and again, have a reduction in TRPM3 function in these natural killer cells from MCFS patients, what confirmed the impairment. So NCNED did a second step that was experiments with in vitro treatment with naltrexone. Because as probably most of you know, ME patients now also long COVID and other conditions, patients use a low dose of naltrexone. So NCNED did repeat the experiment with health control and ME patient cells incubate just as a control. And you can clearly see an increase in current when look health cells, and no difference when look ME. However, in this second group, we applied naltrexone overnight. Health control, as expected, again had the increase of current, and for the first time, we see here cells from ME that present the baseline and an important change with increase of progress amplitude after naltrexone treatment. And when I look all the cells from all patients and compare healthy ones with M patients, cells, there are no differences. What means that the TRP channels in natural killer cells from M patient was restored after incubation with naltrexone. The next step was in vitro experiment. So what NCNED did was compare health control cells with cells from any patient that were taking LDN. So again, similar to in vitro experiment, no difference, compare health ones with any patients taking LDN. So it confirmed the restoration of TRPM3 IO channels in patients that take LDN. So after that, NCNED start to look and ask if are TRPM3 IO channels also impaired, also dysfunctional in long COVID patients. And so we did health controls patient, a new cohort of M patients to confirm previous results, and for the first time long COVID patients. And here, again, the health control is, as I explained before, progressive stimulate, and there are a clear increase in current. However, again, no difference in M patient, and for the first time, we see that long COVID patients also have no increase in current. The doors not open to the influx of calcium when we apply progress. And when you compare the three groups, when compare health ones with long COVID, there are significant difference in TRPM3. The TRPM3 is impaired in long COVID patient. It's also confirmed with EMI, but interesting here, it's clear that there are no significant difference compare long COVID patients and EMI patients. The cells have the same impairment in TRPM3 IO channels. So the second part of record is the close TRPM3, the block of TRPM3, that is the anonymity. And again, similar results, no difference when compare ME, CFS, and long COVID patients. It's confirmed that the cells from ME and long COVID have the impairment in TRPM3 IO channels. So the second step with long COVID was also looking vitro treatment. So here we can see health controls with the increase and clear increase in progress current. And for the first time with long COVID, after in vitro treatment with naltrexone, it's clear that progress will work after a few seconds. And it's here also clear that there are increasing progress current of long COVID patients. So when look all results together, we have no difference between cells from health control and long COVID after incubation with naltrexone. The second, with anonymity confirmed the previous results I just showed, and there is no difference. So how naltrexone work? What is this natural killer cells model? So I'm here again with natural killer cells, with the TRP channels, but naltrexone doesn't work in the TRPM3 specifically. Naltrexone is a mu-opioid receptor antagonist. So what naltrexone does is block the mu-opioid receptor. But it's important for TRPM3 because mu-opioid receptor have the potential to block TRPM3 IO channels. So when I use this drug that block mu-opioid receptors, it release what the mu-opioid is doing with TRPM3, and it will open TRPM3 and make possible calcium influx in the cells, what restores cell function. So what's the next step? Here, I'm talking about this step from NCNED research that I'm directly involved. There are others steps, for example, in Brianna and Kieran are involved and I'm not. So I'm just mention few studies because are the study I'm involved, okay? So the next step is the clinical trials. NCNED are approved to run 2 clinical trials. One specific for long COVID using low dose of naltrexone. This one will commence now, end of May. And the second one with M patient also is low dose of naltrexone that will start in September. What's important here, both are randomized placebo-controlled trial that will use a dose escalation, and both, of course, double-blind, so researchers don't know if patients are taking placebo or low dose of naltrexone, and also the patient doesn't know. And if you are interested in participate, we are in both doing the recruitment. So if you have any interest or know someone interested, please contact Tanya. We provide the email ncned@griffith.edu.au. Also, if you have any question about NCNED research, you can contact us by this email and talk with Tanya, and Tanya can provide information or also give our contacts. Other studies also involve blood donations. So if you are interested in participate, some criteria are the age that should be between 18 and 65 years, no smoker, not pregnant or breastfeeding, and other criteria that excludes are some chronic illness, but please check with Tanya in the same mail. And for studies that involve blood donation, we need between 14 mils and 84 mils of blood, and also that you complete some questionnaires. I would like to highlight here that NCNED also need health controls volunteers. So if you are not able or have any issues with needles, for example, and don't want to participate, don't feel well, you also can inform if you have any relative, family members or friends that are interested. And I'd like to thank you very much all the participants, because NCNED research is just possible because we have collaborators with participants. Without participants, no way to run our experiments and research. And also thank you to all the team and our collaborators and also the fundings. And thanks so much for your attention, and I'm happy to reply questions in the end.